Erection, or the substance abuse and the drug addiction. The substance abuse is a chronic relapsing disorder characterized by compulsive intake of a drug and loss of self control. A drug addiction is defined as compulsive and out of control of drug use despite negative consequences. In substance abuse, persistent desire or craving due to euphoria, pleasure, and reward that the drug produces. The pleasure is produced by the drug by the production of dopamine in a part of the brain called nucleus accumbens. And if the dopamine quantity goes down, the person craves for the drug. This pleasure may be also to food, music, sex, and gambling. What happens in craving that the person goes into a stress when the drug is finished and dopamine levels in the nucleus accumbens falls down. To get that pleasure, the person get into stress. And when there is an stress, the hypothalamic pituitary axis become active, and this is what is the result of craving, in which excess amount of corticotrophin releasing factors are produced to produce cortisol. People with stress have higher level of CRF in CSF. So where is the reward center? The reward center is the region in the basal forebrain, rostral to the preoptic area of the hypothalamus. It is located where the head of the caudate nucleus and the anterior portion of the putamen meet, lateral to the septum pellucidum. There are two parts of the nucleus accumbens, the core and the shell. The dopamine is produced in both the core and the shell, but more in the shell in addicting drugs. So what are the parts involved in the addiction? There are reward center, which includes nucleus accumbens, medial forebrain bundle, and ventral tegmental area. This is the reward center. And also involved are the prefrontal cortex and the frontal cortex, which are concerned with planning and judgment, amygdala, which is concerned with emotions, and the condition effects. The reward centers are connected to each other. This is the basal forebrain, and this is the ventral tegmental area, and here it is the nucleus accumbens, and this is amygdala. Ventral tegmental area has the nuclear projections to nucleus accumbens and to the amygdala, and also to the other parts of the brain, the prefrontal cortex and the frontal cortex, and also to the other parts of the brain. Nucleus accumbens and the ventral tegmental area, they both have inputs and outputs to the different parts of the brain. See here, the nucleus accumbens is sending projections to ventral pallidum, thalamus, and to the orbitofrontal cortex and prefrontal cortex and cingulate cortex, and then from there it's receiving back onto the nucleus accumbens. And here is the amygdala connected back to hippocampus to and fro and paraventricular nuclei, and this is the stress ventral tegmental area. This has dopaminergic neuron, and these dopaminergic neuron This is the most important part of the system reward center. The ventral tegmental area sends projections to nucleus accumbens, to the prefrontal cortex, and to the other parts as well. This is another view of the mesocorticoencephalic dopaminergic system. This here is substantia nigra and ventral tegmental area sending projections to amygdala, nucleus accumbens, into the forebrain, medial forebrain bundle. This is the ventral tegmental area here and nucleus accumbens here. This ventral tegmental area is sending dopaminergic projections to the prefrontal cortex, the nucleus accumbens, striatum, hippocampus, and is receiving glutamatergic inputs from the other parts of the brain. So an addictive drug is a drug is both rewarding and reinforcing. Substance dependence can be diagnosed with physiological dependence, evidence of tolerance or withdrawal, or without physiological dependence, with persistent desire to use, the reduction in social and recreational activities. So the main features of the drug addiction are reward, pleasure, euphoria, and if not present, it leads to craving. So what is tolerance? When the person keeps on using the drug persistently, the number of receptors are decreased. That is, there is down regulation of the receptor. So to produce the same effect, the person has to take an increased amount of the drug. And what is upregulation? The upregulation is that the reverse occurs when the decreased neurotransmitter at the postsynaptic site occur. This results to upregulation with increased number of receptors. The other cause of tolerance is by in the liver, glucuronide transferase system and cytochrome P450 enzyme system. The glucuronide transferase system causes glucuronidation, which converts the drug in a water soluble form so as to be excreted out of the body. If this glucuronide transferase system is influenced by a drug that increases its activity, is called enzyme induction. The drugs that increase its activity or enzyme induction are phenobarb and rifampin. So if the metabolizing system is fast, the drug actions are reduced and they may cause decreased activity. This may be a cause of antibiotic failure and contraceptive failure as well. What is sensitization or reverse inverse tolerance? Sensitization refers to an increased response to a drug effect following repeated use. And one example is decrease in threshold for brain seizure with repeated exposure to cocaine. And what is reinforcement? Reinforcement is the ability of a drug to produce pleasure that motivates to take the drug repeatedly. And what is physical dependence? Physical dependence requires drug to maintain normal functioning. It maintains homeostatic mechanism for normal function. If the drug is stopped, its termination produces withdrawal effect. The withdrawal effects are exactly opposite the drug produces. the depressant becomes hyperactive and the stimulant in a withdrawal effect becomes depressed. But the common feature to both depressants and stimulants is that both of them produce dopamine in the nucleus accumbens to produce euphoria and pleasure. Opioid for pain relief. See, this normal dose for the pain relief causes physical dependence and tolerance, but it does not produce addiction. But a oral, single large oral or parenteral dose of morphine may cause withdrawal effects. So what happens in a withdrawal effect that the depressants like morphine and alcohol, which normally depresses the CNS, but when they are withdrawn, the CNS become hyperactive. And in case of stimulants, which causes stimulation, but in the withdrawal effect, when they are absent, the person becomes depressed. The tolerance, one more point, that can glucuronide transferase induction cause a toxicity of a drug? The answer is yes. If the enzyme induction is present and a prodrug is taken, the enzyme induction converts the prodrug into an active drug faster than normal, and that produces the toxicity. What are the factors involved in the addiction? There are genetic factors involved, and the loci for alcohol are present. Alcohol addiction are present in chromosome 1, 7 and probably on 2. The genetic marker that has been suggested to be indicative of a genetic predisposition to alcohol is related to dopaminergic system. 40% of the alcoholics have genetic predisposition. It is the A1 allele for the gene for dopamine D2 receptor. But the A1 allele has also been associated with abuse of other drugs, compulsive overeating, pathological gambling, ADD, attention deficit disorder, and Trout syndrome. This suggests that A1 allele is not specific for alcoholism. Number two, chronic drug use activates the transcription factor CREB, cyclic AMP response environmental binding protein, and FOS related antigen in nucleus accumbens. Ethanol and amphetamine increase the number and length of dendritic spines in the cortical region. The action of the acetaldehyde dehydrogenase. In the ethanol metabolism, there are two dehydrogenases involved: alcohol dehydrogenase and aldehyde dehydrogenase. Alcohol dehydrogenase converts ethanol to acetaldehyde, and then this acetaldehyde is converted by aldehyde dehydrogenase to acetic acid, and then further metabolism. In some people, especially of the Southeast Asian origin, like Chinese and Japanese people, if this enzyme is slow in action, if the aldehyde dehydrogenase is genetically slow in action, it does not clear acetaldehyde from the body, and that accumulates in the body. This acetaldehyde is toxic to the body, and if accumulated in an excess amount, it produces flushing, headache, nausea, vomiting, and hypertension. So, this is the type of the people in whom aldehyde dehydrogenase activity is slow. They are not prone to develop alcohol addiction, but those who have a fast or a normal activity of aldehyde dehydrogenase, they are more prone to develop addiction to alcohol. The drug disulfiram, which is given to prevent alcoholism, acts at this site. It prevents the action of aldehyde dehydrogenase. So excess amount of acetaldehyde is accumulated causing symptoms. Also, metronidazole causes the same effect of disulfiram like. So the abused drug increases the dopamine mainly in the shell of the nucleus accumbens, but it also increases dopamine in the core of the nucleus accumbens. The nucleus accumbens has 95% of GABAergic neurons which project onto the ventral tegmental area. And the dopamine mainly in the nucleus accumbens comes from the ventral tegmental area, which has dopaminergic projections to the nucleus accumbens. So ventral tegmental area is the main modulator of the reward system. And dopamine receptors are two groups. The D1 receptors activate the cyclic AMP dependent protein kinase by coupling with excitatory G proteins. The D2 receptors inhibit cyclic AMP dependent protein kinase by coupling to inhibitory G proteins. So D1 receptors, they activate the cyclic AMP, and D2 inhibit the cyclic AMP protein kinase. Both D1 and D2 cause drug reinforcement. What is the dopamine transporters? The two drugs, cocaine and amphetamine, they increase the dopamine in the synaptic cleft and produces the stimulation. They have different mechanism of action. Both of them increase dopamine in the synaptic cleft in the nucleus accumbens, but by a different mechanism. This is the presynaptic terminal. This dopamine, it goes back into the presynaptic terminal, and this is the monoamine oxidase enzyme responsible for the dopamine metabolism. Just for a brief review, the end product of dopamine metabolism is homovalenic acid. That of epinephrine and norepinephrine is metanephrine and normetanephrine to vanillin-mandelic acid, and for serotonin it's 5-HIAA 5-hydroxy indole acetic acid. All these substances go back from the synaptic terminal after they act on the postsynaptic terminal, go back for metabolism through the dopamine transporter to be metabolized in the presynaptic terminal by monoamine oxidase. The amphetamine. The difference between amphetamine and cocaine is that Amphetamine goes back into the presynaptic terminal. Its structure resembles dopamine. It pushes the dopamine out of the vesicle into the presynaptic cleft, synaptic cleft, increasing its quantity and causing euphoria and excitement. Whereas cocaine, in this picture, see, this is the presynaptic terminal, postsynaptic area here, where you have the receptor, dopamine receptor. This goes back normally, dopamine, norepinephrine, and serotonin go back for metabolism through the monoamine oxidase system. What cocaine does, that it blocks this uh, dopamine transporter, which is actually a gated channel, and once it blocks, the dopamine cannot go back into the presynaptic terminal, thereby increasing its effects.